Good day, everyone, and welcome to the STEM Lockdown Digital School. My name is Katla Matibe, and I'll be presenting the Life Sciences Grade 10 lesson to you guys. Welcome back to those who have been with us, and platform or joining us for the first time. Welcome. So, guys, do remember to use my email address at ls.wiznet@gmail.com for any questions relating to life sciences, and I will help you guys. So if you need past papers, slides, or any help regarding life sciences, I'll help you. So guys, as you remember, we started off with our new topic yesterday on cell division. And today we are continuing still with cell division, and we will be discussing mitosis. So this falls under the strand life at the molecular, cellular, and tissue level. And as you know, yesterday we did introduction to the cell cycle. So guys, at the end of this lesson, what I want you guys to be able to do is to define concepts that are highlighted in bold black, to understand the phases of mitosis, to differentiate the phases of mitosis in a diagram. So if you are given a diagram, should be able to describe or differentiate the phases of mitosis. And my email address is at ls.wiznet@gmail.com. So if you want it, I'm also typing it out for you guys. So it's under the chat section. There it is, Sophie. All right, so you should be able to understand how mitosis occurs in the plant cell and the animal cell. And you should understand nuclear and cytoplasm division. And lastly, you should be able to know the importance or role of mitosis. So why is mitosis important? Remember, guys, I told you that I am using this section of objectives to guide you guys as to what is important and what you guys should know. So take that into consideration. All right, so just to quickly recap, yesterday we were talking about the introduction to the cell cycle or of the cell cycle. We mentioned that the nucleus contains all the genetic material of the cell. So it is one of the largest organelles in the cells. And remember, the plant and animal cells nucleus are the same. So the nucleus of the plant cell and the nucleus of the animal cell are the same. And we also mentioned that a chromosome is made up of chromatids joined together by a centromere. And I told you guys that this word centromere, you shouldn't confuse it to centrosome and centrioles. All right. So I gave you guys a structure of a chromosome. So you should be able to draw the chromosome and label it. You know that in life sciences, when you are drawing something, when we request you to draw a diagram or anything or a structure of something, you should have a label, a heading. So you should have labels and a heading, and your drawing should be clear and big. All right. Then we also talked about cell cycle, and we said it occurs in 2 stages, which is the interphase and the mitotic phase. So we only dealt with the interphase yesterday. Hence today we are continuing with mitosis, or rather mitotic phase. So during interphase, the cell grows and DNA duplicate or replicate. And we saw from a diagram that we were looking at that interphase takes up most of the time of the cell cycle. So that is what we did yesterday or in our previous lesson. So starting off with today's lesson, we are still talking about the cell cycle. So the cell cycle, as we stated, it is divided into two stages. That is the interphase and the mitotic phase, or the M phase. So this diagram here is what we looked at yesterday. This is the interphase, and it is taking up most of the time of the cell cycle. That is the interphase, and then the mitotic phase. So this cycle differs for both plant and animal cells. So for plant cells, the cycle differs to that of animal cells. So now we are doing the mitotic phase. And the mitotic phase is divided into two tightly joined processes. And that is the mitosis and cytokinesis. So guys, you can see these terms are in bold black, so take note of those. Now mitosis, as we said yesterday, is a type of cell division where one cell divides and it produces two identical daughter cells. Maybe your textbook might say as where one nucleus of a cell divides into two identical nuclei. So mitosis is about the division of the nucleus, and cytokinesis is the division of the cytoplasm. So these identical daughter cells have the same number and type of chromosomes. So these identical daughter cells have the same number and type of chromosomes. Remember we talked about chromosomes? And you saw that how they look like or how they are. So they have the same number and type of chromosomes. And these are similar to the parent cell. And mitosis is then followed by cytokinesis, in which the cytoplasm, organelles, and cell membrane split into two cells containing roughly equal shares of these cellular components. So cytokinesis has to do with the cytoplasm, the division or the splitting of the cytoplasm into two. And when it splits into two cells, both cells contain equal shares of these cellular components. So this is it then divides or splits into two. So as it splits into two, we get daughter cells that are equal and have the same number and type of chromosomes. So that is what mitosis is. So mitosis has four broad phases which occur in it. Firstly, it is the prophase. Then we have the metaphase, and then the anaphase, and lastly the telophase. So these phases will be explained in detail individually as follows. So the first one is the prophase. Now, when does the prophase take place? When does the prophase take place? When does the prophase take place, guys? So the prophase takes place immediately after interphase. So immediately after G2 phase of the interphase. So prophase immediately after the interphase or G2 phase of the interphase. And we said the interphase is when DNA in the chromatin network duplicates, thickens, and becomes visible. Then we get the prophase. So this is the first phase of mitosis where the nuclear membrane and the nucleolus disintegrate. If so, in animal cells, the centrioles separate and move to opposite poles. Remember we talked about centrioles when we were doing the organelles of the animal cells? So in animal cells, the centrioles separate and move towards the poles, opposite poles, and these centrioles give rise to spindle fibers which form between the poles. Then in plant cells, there are no centrioles. Remember, plant cells do not have centrioles. Therefore, in plant cells, spindle fibers form in the cytoplasm. So there is nothing, there are no centrioles at the poles of plant cells. Therefore, spindle fibers just form in the cytoplasm. So this is what we are talking about. This is an animal cell, and these are your centrioles. So they are moving towards the poles. These are the poles. Or if you prefer this image the other way around, then your poles would be here up, and at the bottom there is your south pole. So centrioles are moving to the poles, and they have spindle fibers. So these lines here are your spindle fibers, and this is the nucleus. Remember, the nuclear membrane is this integrating, or it is disappearing. So as it disappears, the chromosomes now become visible from the interphase. Remember, the interphase is when your chromosomes become visible. And they are doing that because the chromatin network untangled. Remember, I told you guys that it looks like noodles. So it untangles, then the chromosomes become visible due to DNA replicating and the cell growing. So as these centrioles come to the poles, the spindle fibers then form and connect to the poles. This is for animal cells. They have centrioles or centrosomes. And then for plant cells, there is no centrioles, as you can see, and the spindle fibers. So these are the spindle fibers. It looks like wool, something. So these are your spindle fibers. So it is a micrograph. So they are connected to the spindle fibers come from your cytoplasm. Then you have your spindle fibers. Now, as you can see, these are your chromosomes. And also for the plant cell, the nuclear membrane is disappearing, similar to that of the animal cell. So it is disappearing. Now that is prophase. So in prophase, animal cells, centrioles move to the poles, towards the poles, and spindle fibers then form. And for plant cells, because it does not have centrioles, spindle fibers just form from the cytoplasm. and we normally use the word condense. So maybe in your textbooks, you can see the word that I just mentioned now, condense. So the chromatin network then condenses. So it condenses as the nuclear membrane disappears. So the chromatin condenses into chromosomes, and the nuclear envelope or membrane disappears or breaks down. In animal cells, the centrioles move towards the poles, and in plant cells, there are no centrioles. Spindle fibers just form from the cytoplasm. And that is your prophase. Then we have metaphase. So during metaphase, remember, you can remember this by saying M for middle. So during metaphase, the chromosomes line up on the equator of the cell. Hence I said M for middle. So the chromosomes then line up in the middle of the cell, or the equator of the cell. Or your other textbooks might call this equator the metaphase plate. So it is also called the metaphase plate. So during metaphase, chromosomes line up on the equator of the cell, and these chromosomes appear in a straight line across this middle center, or the middle of the cell. Now, the chromosomes are attached to the spindle fibers, it is in bold black, by its centromere. So it attaches the chromosome. Remember, the chromosome has a centromere. The centromere joins two chromatids together to form a chromosome. So these spindle fibers from the centriole attach to the chromosome by its centromere. So they attach at the center. That center called centromere of the chromosome. So this phase is the same for both plant and animal cells. So as you can see in plant cells, they are also lining up in the middle of the cell, and the spindle fibers, which are connected to the cytoplasm, then attached to the centromere of the chromosomes, which are lined up on the equator of the cell. So remember, first letter M from metaphase. So remember that M for middle. And then we have the third phase of mitosis, which is anaphase. So during anaphase, the chromatids are pulled to opposite poles of the cell by shortening the spindle fibers. So during anaphase, chromatids are pulled apart. So remember A in anaphase for apart. So these chromatids, remember chromosome is made up of two chromatids. So the chromatids are pulled apart towards opposite poles of the cell by shortening the spindle fibers. So this is what I am talking about. You have your centriole here for the animal cell. Spindle fibers are shortening as the chromatids are pulled apart. So there's a chromatid, there's also a chromatid. So the chromatids are now called single-stranded daughter chromosomes. They are now called single-stranded daughter chromosomes. And the process is also the same for plant cells, as you can see on this diagram here. Spindle fibers are becoming short as the chromatids are pulled towards the poles. So that is anaphase. Then we have telophase. So during telophase, a nuclear membrane reforms around the daughter chromosomes that have gathered at each pole. So these daughter chromosomes uncoil to form the chromatin once again, and the nuclear membrane reforms. So the nuclear membrane reforms. Remember, it had disappeared, or it had disintegrated, or it was broken down or broken up during prophase. And the chromatin network condensed into chromosomes so that chromosomes become visible. So now for telophase, it is the reverse of that, the opposite of prophase. So for telophase, the nuclear membrane then reforms, but now we have two cells. It then reforms for both cells, and then the chromatin network is then formed again. Then we end up with two identical daughter cells. So these daughter cells are identical to the parent cell by what? By them having the same number of chromosomes and same type of chromosomes. So this process is the same for plant cells also. So the nuclear membrane is now reforming around the daughter cells that have gathered at the poles. Then we have two cells. So guys, mitosis is quite easy. Remember these steps. And then we have cytokinesis. So cytokinesis, remember we said the M phase is divided into two stages, which is the mitosis, or two processes rather, which is the mitosis and the cytokinesis. So cytokinesis, the cytoplasm then divides during a process called cytokinesis. Remember we said cytokinesis, the splitting of the cytoplasm. So the cytoplasm divides and splits into two. So in an animal cell, the cell membrane constricts. Folding of the cytoplasm divides the cell into two. So for animal cells, the cell membrane constricts. Remember, the animal cell does not have a cell wall, but a cell membrane. So the cell membrane constricts, and this enfolding of the cytoplasm divides the cell in two. So we then have two identical daughter cells. But in a plant cell, a cross wall is formed by the cell plate dividing the cytoplasm into two. Sorry about that. So a cross wall, if you look here, is formed by the cell plate dividing the cytoplasm into two. And then we have two genetically identical daughter cells, which are identical. Process starts again. So from these two, how from these two daughter cells. So after mitosis occurs again, how many new daughter cells will we have? Remember, these two daughter cells will then be the parent cells. So how many will we have if we have these two? I don't know if I should choose like we did yesterday. I unmute you, you answer me, or you use the chat box or chat section to answer. So Nkosipila says 4, Israel 4, Tyrese 4. Yes, guys, we will then have 4 new cells. We'll then have 4 new cells. Thanks for the responses. So that is all. That is mitosis and cytokinesis. So it starts off with interphase, where the cell grows and the DNA duplicates. Then we have prophase. Then in prophase, the nuclear membrane disintegrates and the chromatin network condenses. So that is when we see our chromosomes. Then we have metaphase. Now this phase, the chromosomes then line up in the middle or at the equator of the cell. Then we have anaphase. So anaphase, I say you can see the letter A. Remember, it means apart. So then the chromatids are pulled apart to the opposite poles of the cell. And then for telophase, the nuclear membrane then reforms and the cell then divides into 2. Then cytokinesis, it is just the splitting of the cytoplasm into 2 to form the 2 identically, or rather genetically identical daughter cells. So by genetically, we mean that they have the same number of chromosomes and the same type of chromosomes. Now, guys, you know that we have different genes, right? So people have different genes, so our genes are not the same. What about twins? Do twins have the same genes? Do twins have the same genes? So Tyrese says similar and not the same. And also Letabo says almost. Okay, so almost the same. Ngosipila says yes. Lely, not completely the same. But why do you guys say they do not have the same? Or why are they not similar? Or why are they not the same, but similar? Tyrese says there's always a difference. What kind of a difference, Tyrese, between the twins? All right, so what about identical twins? Because Tyrese says the looks they may look different. So what about identical twins? So when we talk about identical twins, do they have the same or identical genes? So Lali says with identical twins, their fingerprints are not the same. So no, it's similar. Thank you guys. So twins have similar genes and not identical. So you guys get this. This is awesome. And what do What is the role of mitosis? What is the role of mitosis? So if it is cell division, why do we need many cells in our body? As human beings, why do we need many cells? Why do we need replication of cells? Is our cells for growth? Yes, we need to grow. So all another reason. So what happens as we grow? So Tyrese says mitosis is important for creating more cells that are genetically the same. But for what purpose? That is correct. So Tyler says repair cells and to replace these cells. So they need to be replaced. So an example, give me a everyday life that you might think of. An example of mitosis in our everyday life. In our everyday lives. So I'm going to unmute some of you. Give me the answer. So Tyrese went silent yesterday. I'm not going to unmute you again today. So Tyler, an example of mitosis in our everyday lives. Hi, Tyler. All right, so Tyler is doing exactly what Tyrese did yesterday. So, okay, you don't know one. So anyone, give us an example. You guys are saying mitosis is needed for growth. To repair cells and to replace cells. So Tyrese is saying skin. So you explain to all of us what you mean by that. What you mean by blood and skin.
Yo, ma'am, you see, so like mitosis almost like produces cells.
Yeah.
For the human body. So that's what I'm saying. Like these cells include like skin cells, blood cells,
On blood cells. Oh, okay.
or maybe even No, not muscle. I think muscles also, man. I'm not sure though.
All right.
Yeah, that's my answer, ma'am. Ish, sorry.
No,
Sorry
it's
about
okay.
yesterday, ma'am.
It's all right, Tyrese. Thank you for your answer.
Alright, no
So
problem.
Tyrese says for skin and the blood cells. So when you get a cut, you bleed, and then suddenly the blood clots. That is what Tyrese means by blood cells. So the blood clotting, it's a form of mitosis. All right. So your blood clots to stop the bleeding and to replace the cells that are lost when you bleed. So that is basically mitosis. And Tyler says reproduction. I'm doubting reproduction, but how? AL says you replace body parts. Yes, you replace body parts. So if you get a cut, your skin then joins again. Or if you go for surgery, you get stitches, they stitch you. After some time, your skin is now joined. It's because of cell division. So you guys are on the right tracks. Thanks for these responses. So functions or the roles or the importance of mitosis is for growth. So in multicellular organisms, mitosis is needed or cell division is needed for growth. So they all start as a single cell and soon have a huge number of cells. And then you mentioned repair. So organisms constantly repair and renew themselves. So worn out or dead cells are replaced through cell division. And number 3, reproduction. I'm going to give you the answer, Tyler, because you just said reproduction. So single-celled organisms such as bacteria and protists also reproduce by cell division, and that is called binary fission or budding. So there's an example of a single-celled bacteria or protist undergoing the process of budding. So this is what they do. It was one bacteria and then it underwent reproduction. Then as it buds, it produces 2 bacteria cells. So this is the importance of mitosis. Those examples you guys can give out. IPMAT to remember the phases of mitosis. So IPMAT, so IPMET for the phases, to remember the phases of mitosis. What are some of the ways you guys remember these phases? Because I'm suggesting IPMAT. So what are some of the ways that you guys prefer to remember these phases? Or how do you remember them? So I is for interphase, P prophase, M metaphase, A for anaphase, and main T is for telophase. Zach gave us this yesterday. So I see Zach is absent today. So Zach gave us this acronym yesterday. So Nelly says, I play my accordion today. That's how Nelly remembers the phases of mitosis. I play my accordion today. So I for interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase. It's very smart, Tyrese. Very smart. So what are other ways? Remember, by sharing, some of your classmates may use those or your suggestions or your abbreviations or acronyms because they might find them easy. So you are helping others. So you can remember these in many ways. So guys, this is your homework. Between plant and animal cells during the process of mitosis. Remember we said the cell cycle differs for plant cells and animal cells. So you guys can tabulate the differences. We talked about those in our slides. Yes, Tyrese, tabulate is make a table. So you can make a table, have plant cell as your heading, or on another column, and then animal cells on another column, then you give out your differences. They can be 2 or 3 if you have 3, but 2 is enough. So the takeaway message, guys, the process of mitosis occurs in 4 stages, that is prophase, metaphase, anaphase, and telophase. And then we have cytokinesis, and it differs in plants and animal cells. We said that in animal cells, the cytoplasm invaginates or enfolds, and it divides the cell in 2. And in a plant cell, the cell is divided into 2 by the cell plate, which forms the cell wall. And then the functions of mitosis, it ensures growth of tissues and organisms, and damaged and worn out tissues are repaired and replaced by new cells through mitosis. And lastly, single-celled or unicellular organisms like the amoeba often reproduce asexual reproduction on its own. So it reproduces on its own. So guys, these are some of the references I used. The diagrams are from the Siavula textbook, which is free online. And also the Via Africa textbook is also free online. Some of the helpful links. So guys, tomorrow we are going to do some exercises on mitosis using Kahoot. So you guys, if you have 2 devices, then you are good to go. We are going to use Kahoot for our exercise tomorrow. I'm going to do exercises on mitosis. So do remember to follow us on all social media platforms. So subscribe on our YouTube channel at Africa 10 Gigs, and you guys will be able to see all these videos if you missed out and want to catch up. Our Facebook page, Instagram and Twitter at Africa 10 Gigs. See you guys tomorrow, same time, same place, and thanks for a great lesson that we had.
Okay, bye, ma'am.
Bye.